Welcome to my YouTube channel, and this is Ricky Chisambo. In this lesson we want to revise biology paper 2, which was written in 2018 in July. This is examination for General Certificate of Education Ordinary Level. So this is the GCE exam, and in this paper we'll look at section A. So section A is short answer questions, which carries 44 marks. So the first question is, figure 1.0 shows a diagram of a developing insect. So when we look here, we have this is an insect. So now you'd find that how can we know that this is an insect? Number one, the characteristics that shows that this is an insect is that it has been divided, or it has the body is divided into three parts. So now when you look at an insect, so we have different parts which have been labeled here. So now we can look at the question. From figure 1.0, identify three parts which shows that the insect is a living organism by associating the parts to labeled particles, characteristics of living things. So meaning that this is the first topic that you learn in grade 10, the characteristics of living organisms. How can you know that this is a living organism? So number one, you'd find that we have number one, we'll write it M-R-S, and then we have G, E, and N. So you'd find that they make characteristics of living organisms, where we say this is movement. So meaning that one of the characteristics of living organism is movement, and then the other one is respiration, and then sensitivity, and then the other one is growth, and then the other one is excretion, and then N is for nutrition. So nutrition, N is for nutrition. So this is Mrs. Green. So this acronym, which is Mrs. Green, will help you to know the characteristics of living organisms. So now when you look at the question here, they've said from figure 1.0, identify three parts which shows that the insect is a living organism. So a living organism associated with these characteristics that we've mentioned here. So we can look at the insect given. So the insect given here, the insect which is given here, we have to know and identify the parts. So now we have part 1. So from the insect, part 1 is we have antenna. So antenna, you'd find that the antenna helps the insect to, number one, or to see, or for sensitivity sake. So that is the first part that we can look at. The first part is the antenna. So the antenna characteristics of living organism, from these that we've mentioned here, you would find that it is sensitivity. So it is sensitivity. Okay, so let us look at the second one. So the second one, when we look at the second one, we can look at the second one is that we have wing. So wing here. So now wing, when we look at wing, what What is the function of these wings? So the function of these wings is that they help the insect to move. So we've identified the second one, which is the part wing, and then for wing is movement. And then the third one we can look at, let us identify the third one. The third one, yes, we have this, the third one, where we have mandible. So this one, it helps the insect, or it helps the insect to grab any food substance using the mandible. So meaning that if they grab, they take in. So this is for nutrition. So when you look here, we have so mandible for nutrition. Okay, so we can move. State 2 other characteristics of living things not shown in figure 1.0 above. So we have to know and identify. Let us see what is not here. From these characteristics that we mentioned here, what can we see which is not included here in the figure given? So the first one that we can observe here is that there is excretion. So excretion, we can't see anywhere where the insect is excreted, but you'd find that because this is a living organism, meaning that there will come at a time after it has taken in food, so obviously at some point it should excrete. So meaning that one of the characteristics not mentioned or not shown here is excretion. So we know excretion is the removal of metabolic wastes. So each and living organisms, they excrete, meaning that they release metabolic waste, those wastes or the byproducts in short. So the second one here is, what can we see from the characteristics given? So from the characteristics given, we haven't seen where they've said, because we know at some point this insect had to grow, and then what else? Apart from that, we've seen sensitivity. Now respiration, we haven't seen it, so you'd find that it is not included here, but it helps, or it is one of the characteristics of living organisms. So meaning that we have respiration. Okay, which characteristics of living things is only found in animals? So which is found in what? In animals. So you'd find that these characteristics that we mentioned, which are 7, so these characteristics that we mentioned, you'd find that in plants, so in plants and animals, so you'd find that both there is growth. So both there is growth, and then both there is respiration, and then what else apart from that? In both there is excretion, and then what else apart from that? You'd find that the only difference that we can see here in plants and animals is number 1, locomotion. So locomotion, it is one of the examples that we can give there, or we can look at sensitivity, and then or we can look at movement, meaning that the plants, they can't move from one place to another. So the plants are static, meaning that once it grows, unless you want to plant it somewhere else. So you'd find that the answer that we can give here, it is so locomotion. That is the answer that we can give. Okay, so we can move. What are the two processes which makes up metabolism in living organisms? So we want to look at two metabolisms that are in living organisms. So we know that there is number one, so catabolism, and then the other one it is anabolism. Okay, so these are the answers on question C. So we can move to question 2. Figure 2.0 shows a protein molecule undergoing digestion in the alimentary canal of a human body. So you'd find that these are proteins. So these are proteins, and then we have the catalyst, which is reacted by what? By K. And then here we have the products, and then so when you look at this, we have the reaction, which is M there. Now, without wasting time, we can go to the first question, which is identify the products labeled L. So we want to know the product which is labeled L. So the product which is labeled L. So meaning that once this has been produced, and we have two, so here this one and this one, once they are produced, they are going to produce what? L here. So now when we look at this, you find that these two, they can be amino acids. So this one, it is amino acid, and this one, so meaning that the two, they will make what we call peptide. So peptide, it is the combination of two amino acids. Okay. Suggest a region in the alimentary canal where reaction K takes place. So we want to know where reaction K, this reaction takes place. So where exactly? So you'd find that because these are proteins here, the product here, they are proteins. So where do this reaction takes place? So because we are looking at the alimentary canal, that is, so suggest a region in the alimentary canal where reaction K takes place. So the answer here, it is stomach. Okay, reaction M completes the digestion of proteins. So we have reaction M, which completes the reaction. So this M, it completes what? The reaction. Now when we look at the question, so reaction M completes the digestion of proteins. So which enzyme catalyzes reaction M? So we want to know which enzyme catalyzes reaction M. So the enzyme is, we're going to look at, when we look at this one, which is dipeptide, so, or dipeptide, meaning if we just add to this SE, it means that that is an enzyme, that is what will be produced here. So meaning what we're going to have is dipeptides. Okay, so that is an enzyme which will be produced. Okay, so here we are looking at, because we are looking at the pH at which reaction M would be catalyzed by the enzyme identified in B1 above. So meaning that when you look at the pH here, so the pH will tell to say, so the pH will tell us to say, number one, the pH, it has two sides. So which tells the acidity and the basicity of the substance. So here we are looking at which, so meaning we haven't been given exactly at which temperature did the enzyme reacted. And then here what we have to know is just the one side, so it can be the acidity or the basicity. Now we cannot see the basicity, we cannot say the basicity, or we can use the word the alkaline. Suggest where the enzyme named in B1 above is produced. So it is produced in the jejunum. Identify the products labeled N. So we have to identify the products labeled what? Labeled N. So the product labeled N. So these product which here, or the product labeled what? Labeled N. So these are what we call amino acids. So amino acid, we are saying that this is a building block, or it is a building block for proteins. So C, describe 3 adaptation which makes the ileum suitable for digestion of food. So we have to look at the ileum here. So now we are, how do the ileum suitable for digestion of what? Of food. So the first one that we can say is that the ileum is very long to produce a large surface area for adaptation of food. That is the first one that we can look at. So we are saying that the ileum is very long to produce, so to produce large surface area for absorption of food. Okay, the second one, so apart from the first one here, the second one, we are saying that it has a thin line for efficient diffusion of digested what? Food. So it has a thin line, or we can say epithelium for efficient diffusion of digested food. Okay, so the third one and the last one, we are saying that it has villi and microvilli to further increase the surface area for absorption of digested food. So it has villi and micro to further increase for absorption of digested food. Okay, question 3. Figure 3.0 shows part of urinary system. So we have the urinary system. So when we look at this, you'd find that this is the diaphragm there, and then we have O, and then we have P, and then we have Q, and then we have R. So we have to identify these parts. So when you look at, first of all, when you look at O here, this is a blood vessel. So this is a blood vessel which carries blood, and then from there we have kidneys, and then this is apart from kidney, these are kidney. So here we have, and then this is a blood vessel. Now we have to know this blood vessel. What is this blood vessel? So this blood vessel is the vena. So vena cava, and then from there we have, this is the ureter. So this is the ureter because it carries urine from the kidney and then up to the bladder here. So let us look at here. R is urinary bladder. Okay, so let us see the questions. So name the part labeled O, Q, and R. So we've labeled these parts to say this is the vena cava, and then apart from that we have ureter, and then from there you have the urinary bladder. So bladder stores urine. So ureter, it is a tube which, or it is a passage through which urine passes from the kidney to the bladder. Suggest 3 substances excreted by excretory organs shown in figure 3.0. So meaning that we have excretory organs. So once we hear this, you find that excretion, this is the removal of metabolic waste from the body of a living organism. So the main function, or the main function of these excretory organs is to excrete, to remove those wasteful substances that are not needed in the body. So now you'd find that there are several substances that are released and that are excreted. So in an event that we are looking at the question like this one, so there are 3 main substances that are excreted from the body. The first one, when we look at urine, so urine comprises of, number 1, urea. So urea, it is the major, and then apart from that we have mineral salts. Mineral salts, they are excreted also, and then we have excess water. So excess water is also excreted. Once you take up a lot of water, and then now the body will balance the amount of water which should be left in the body. So a volume of water will be excreted because it might not be needed by the body at that time. So give one example of osmoregulatory function of the kidney. So we want to look at osmoregulatory function of the kidney. Now when we look at the word osmoregulatory, so osmoregulatory, it comes from osmoregulation, meaning that it is the body will maintain the volume of water which is in the body. So in an event that you have a lot of water in the body, so this kidney, or the kidney, will help to maintain the normal volume of water which should be found in the body. So now, apart from that, we can look at one example of an osmoregulatory function of the kidney. So we're looking at water there. So now you'd find that the kidney, what they do, once the water is being excreted and they are stored here. So you'd find that they are stored here in the kidney. So in an event that you run out of water, the same water which has been excreted will be reabsorbed in the body. So will be reabsorbed in the blood system. So meaning that the kidney will just store water that has been excreted, and then in an event that you are in need of this water, so water will be reabsorbed in the body. So we are saying that the reabsorption of water in glomerulus filter or filtrate with aid of the hormone which is ADH. Okay, identify 2 disorders that might affect the excretory system. So we are looking at those infections or those diseases that may affect the excretory system. Initially, those that will go direct and affect the kidneys. What are those diseases or disorders that will affect the kidney? Number 1, you'd find that if a person has high BP or BP, you'd find that will lead to destruction of the kidney. So high blood pressure, so high blood pressure. So a lot of people, they have this blood pressure, so meaning that this can also lead to the damage of the excretory organs. Apart from that, we have the other one, it is diabetes mellitus. So that is another that will affect. So don't say that accident. So accident, it is not a disorder or a disease. Okay, so we can move. Okay, so figure 4.0 shows the vertical section or vesicular system in a dicotyledonous stem. So this is a dicotyledonous stem, meaning that a di, when we look at di means what? Means 2. So now, this is a stem here. So in the stem, you'd find that we have the cambium, which is here, and then we have S and then T. So when you look at the arrows, there are arrows that are coming down from the top, and then so we can just say top and then top, and then here we have, so this is the bottom. So we have the arrows that are coming from the top coming down to the bottom, and then we have the arrows that are going up. So we have these arrows that are going up. So arrows that are going up. So we have to, no, because this is a stem, and then we have these arrows that are coming down here. So we have the arrows that are showing that obviously the movement of substance is here. Now let us look at the question. So identify structure T. So we have to identify structure T. Now we have to be very careful when identifying the structures here. So when you look at the structures here, the structures, so we have number 1, these are moving up. So food substances are being moved from the bottom to up, and then we have from the top to the bottom. So we have to identify T. So when you look at T, so this is a cell which has been specialized. So now there are two important cells here. So we have the xylem and the phloem. So now when you look at the arrows which are showing that obviously substances are coming down from the top, so it can be from the branches or from the leaves coming down to the stem or and coming down to the roots. So what can be this structure which is T? So there we have, so the answer is the phloem or phloem, sorry, so phloem. And then what two features in the diagram helps you to identify T in A1 above? So we have to know and understand what made us to identify that this is the phloem there. So now when you look here, though it is not very clear, so when you look here, so we have other cells. So these cells, you find that these cells are what we call the companion cells. So these are companion cells. That is the first one that helped us to identify the answer given. And then apart from that, we have the plates here. So again we have the other plates which is in the middle of the cambium there. So those also, this part also helped us to identify the part. So we have the plates there. So these plates, they helped us to identify. So we can say the presence. So the presence of these plates. So now we can write to say, number one, the presence. So the presence of the companion cells. And then here we have presence of those plates. Okay, name two substances transported by S. So we have to know the substances that are transported by S. So meaning that they are moving from the roots going to the top. And then those substances that are released or that are absorbed from the roots, and then they will go to the upper part of the plant. So those are, number one, we have water, and then number two, we have mineral salts. On the diagram, label a companion cell. So we have to label the companion cell, or lucky enough we've labeled one. So we can say this is C1. So we can say C1, which is companion cell. Okay, so that is C1. We've labeled C1 there. And then explain the role of the companion cell in the vesicular bundle. So we have to know, so they control the activities of the phloem vessel by producing enzymes. So meaning that these they help, number one, in not only they help in controlling the activities of the phloem, but also you find that they help in locomotion to give the position or the structure of the stem. So that is the role of companion cells in the vesicular bundle. So we are saying that it controls the activities of the the phloem vessel by producing okay. Apart from that, explain the function of the cambium. So we want to know the function of the cambium. So the cambium has been labeled. When you look at here, the cambium has been what? Has been labeled. So this is the cambium. So cambium, mostly it is found in the middle of the stem. So now the function of the cambium. So the function of the cambium is that it repairs damaged tissue. So that is the first one. So it repairs damaged okay. And then the second one is that it causes secondary growth. Okay. Okay, Figure 5.0 shows inheritance of red-green color blindness in generations of 2 families. So Y and Z. So we have family Y and family Z. So here we have red color blind male. So meaning that this one here is, so we have this one is a male who is have this disorder, and then we have another male. So meaning that the females were okay, and then here the parents were okay. So the parents were okay, were fine, and then they had to produce children who were color blinded. And then here family Y, you'd find that the man was having this problem of color blindness, and then he married someone who is or who doesn't have this. And then the children that they had to produce here, they are all free from color blindness. And then you'd find that someone who is a female normal from this family married to a man who is color blindness from family Z, and then they had to produce these offsprings. So here we have this one who is a female. So here we have a female who is color blindness, and then we have a female who is normal, and then we have also a male who is normal, and then we have a male who is color blindness. So now let us look at the questions. So which letter in figure 5.0 corresponds to a second folio generation offspring? So we have to look at which one here. So which letter in the second? So in the second folio generation offspring. So because they've said a letter, so meaning that this is the second generation. So this is the second generation. So you find that this is the second generation because they are produced, or they came from the children of family Y and Z. So here the answer is, so because they've said a letter, so meaning that the letter is L. Apart from that, we can move to B, using symbols X. So which is, this is a normal. So this is a normal, and then we have the X, small letter R, which is we have a carrier here. So determine the alleles for red-green color blindness and for the normal sight from the pedigree diagram in figure 5.0. Red color blindness, so we have to know red color blindness. So red color blindness. So for red color blindness. So when you look at here, so we have, this is a male. So this is a male. We can write to say, so male is X, Y. So meaning this one is color blindness. And then here we have a female who is normal. So we can say a female who is what? Who is normal here. So a female who is normal here. And then we may not know if at all the female was a carrier, but we are going to determine that. And then from this also we have, this one is also a person who is affected by this disorder. And then here you'd find that this one, obviously it is R. And then we have also these, they were normal. And then obviously this one, there is a possibility that this one was a carrier. So this one was a what? Was a carrier, because it came from, so we have, it is like this. So this is the father. So now the mother was okay. So the mother was okay. So it can be like this. So where the mother is okay. So once you find that maybe this one was produced, so we have that is, this is what we are looking at. So meaning that the mother or the father had to pass this allele to the daughter, or to, yes, to the daughter who is the female. So now when you look at the question here, the verse has to say red green color blindness. So red, we are saying that it is, we have to write, okay. And then normal sight, so normal, okay. So predict the genotype of offspring J and K. So we have to predict the offspring of K, J and K. So we have J, where is J? So we have J here, and then we have K. So we can predict this. So we can predict this for J, and for what? For K. Now when you look at J here, J is female. So meaning that this is what we are looking at here. So this is what we are looking at here. Or I write it properly to say that is a normal. So a normal female, but she can be a carrier of color blindness because of the father. And then here at K, who is the male. So because here it is direct, they've said green color blind male. So here it is very simple. We can say that is, okay, so let us write. So the first one is, then J is female. So J is female. So we are saying that, okay, so she was a carrier. And then genotype for K, so K is male. So this one had color blindness. Using the genotypes of offspring J and K in C above, show using a genetic diagram the inheritance of red color blindness of offspring L and M. So this one is very simple. This one is very simple. So you'd say the first thing that you have to start with is the parental phenotype. Okay, so normal, I would say normal vision, female. And then we have red, green, color, so blindness, male. And then we can move, so this is female, and then this is male. Okay, so we can move to the second one, which is the parental genotype. Okay, so here we've said she was normal, so, and then here we have, so he was an affected person, and then we can look at the gametes. Okay, we can separate them, so I would say, and then here we can separate them as well. Okay, even here, and then same applies there. Okay, from there we can look at now the parental genotype. So this one will go with that one, so it will be okay, and then this one will go with that one, so here it is okay, so we have this one goes with this one, and then also that one goes with okay. So now we can write to say parental, oh sorry, this is okay, we can say these are offsprings, so here these are offsprings, so we made a mistake here, so these are offsprings, so these are offsprings, you should be very careful. So these are offsprings of parental genotype, so meaning that we have offsprings. Okay, so offsprings phenotype. Okay, so here this is normal, so normal female, and then here we have normal male, and then here we have color, so color blindness female, and then here we have color blindness male. So here we are going to say, so meaning that when you look at here the question, okay, so L, we have L and what, so these are normal, so they are normal, so let us identify who we are normal here. So we identify those that were normal, so those that were normal, so we have female who is L, and then we have M male there, and then this is, you can write this is F2, and then this is F2 there, and then we have F1, so this is F1. Okay, so it means that you've gotten the marks, which is a 4 marks there. Thanks for watching this video. If you are new, you can kindly subscribe for more videos.